Assalamualaikum and hello everyone. Today our group will present the topic which is the simplest invertebrate. So now let us start with sponge. Sponge are the only animals to lack through tissues and have only a cellular level of organization. The characteristics of sponge. Sponge have only a few cell types, like the nerve and muscle cells seen in more complex animals. The circular body of a sponge is perforated by many pores. Sponges are aquatic, largely marine animals that vary in size, shape, and color. This is the sponge anatomy. The wall of a sponge contains two layers of cells, the outer epidermal cells and the inner collar. Collar cells have flagella that beat, moving the water through pores. Food particles in the water are trapped by the collar cells and digested within their food vehicles. This is the canal system in sponges. Sponges have a canal system or spores of varying complexity that allows water to move through their bodies. Interior of canals is lined with flagellated cells that resemble conoflagellates. The cells are called collar cells or conocytes. The skeleton of sponge provides structural support for its body. All sponge contain tough fiber made of spongin, a modified form of collagen. Endoskeleton of a sponge contain small needle-shaped structures called spicules. They are bony, glass, and spongin sponge. This is the difference between sponge and spicules. Sponges fibers that give the sponge its shape and keep it from collapsing, while spicules is the skeleton made of tiny calcium carbonate or little spikes of silicon dioxide. Sponge can reproduce both asexually and sexually. Asexually, reproduce asexually by fragmentation or by budding. During budding, a small protuberance appears and gradually increases in size until complete organism form. Budding produce colonies of sponge that can become quite large. For sexually, during sexual reproduction, eggs and sperm are released into the central cavity, and the zygote develops into a flagellated larva, which may swim to a new location. These are the picture for asexual reproduction and sexual reproduction.
Now let's move on to Ctenophora. Ctenophora, also known as comb jellies, can be found mostly free swimming in warm water. They are propelled by beating cilia and have a range in size from few centimeters to 1.5 meter in length. Other than that, Ctenophora's body is made up of a transparent jelly-like substance called mesoglia. They also capture their prey by using sticky adhesive cells called coloblasts. Some ctenophores are bioluminescent, meaning that they are capable of producing their own light. They use cell division to replicate and regrow their muscles and tissues if lost. Not to be missed, earlier I mentioned about coloblasts, a sticky adhesive cells. Coloblasts are unique multicellular structures that found in ctenophores. They are widespread in the tentacles of these animals and are used to capture their prey. Now let's talk about cnidarians. Cnidarians was placed as a group with ctenophores on the animal phylogenetic tree evolving after sponges. Cnidarians, or we can call it as phylum Cnidaria, is an aquatic invertebrate animal. It is bell-shaped and radially symmetrical that reside mainly in shallow coastal waters. Cnidaria is derived from the presence of specialized stinging cells, cnidocytes. Each cnidocyte has a nematocyst, a fluid-filled capsule, while nematocyst contains a long spirally coiled, hollow thread and it will discharge when the trigger of cnidocyte is touched. Some threads might merely trap a prey or have spines that penetrate and inject paralyzing toxins to its prey. Cnidarian has two-layered sac body that's separated by mesoglea, outer tissue layer and inner tissue layer. Outer tissue is a protective epidermis derived from ectoderm, while inner tissue is derived from endoderm. Inner tissue secretes digestive juices into gastrovascular cavity that also serves as supportive hydrostatic skeleton. Inner tissue also functions in the food digestion and nutrients circulation. There are two basic body forms of cnidarians, including polyp and medusa. There are a few differences in these two bodies. For example, polyp's mouth directed upward while medusa's directed downward. Medusa has more mesoglea than polyp. Next, polyp is a sessile organism, make it fixed in place, while medusa is a free-swimming organism. Polyp reproduces asexually by budding, while medusa reproduces sexually by gametes. Here are some diversity of cnidarians: Portuguese man of war, cup coral, hydra, sea anemone, and jellyfish.
Next, I will explain about a typical cnidarians that are used to study about cnidarians, that is the hydra species. It is usually found at lakes and ponds, usually attached to underwater rocks and plants. Its characteristic, it is small and has the shape of tubular polyp. It has one opening that acts as an anal and mouth. The hydra's mouth is also surrounded by tentacles, and it has a large number of nematocysts. Hydra reproduce by two ways. This is asexually and sexually. Under favorable condition, Hydra will reproduce asexually, that is called budding. Hydra will produce a small outgrowth Hydra from the parent or mature Hydra. Then, the small Hydra will develop and detach from parent Hydra when mature. From this picture, we can see that a small Hydra is start to develop from a mature Hydra. Next, under unfavorable condition, Hydra will reproduce sexually. That external fertilization will occur, where the sperm from testes of male Hydra will swim to an egg of an ovary. An embryo will form and will be encased in within a hard protective shells. And when the condition is favorable, it will emerge and develop into new polyp, which indicates a new hydra. So this is the microscopic anatomy of hydra. There is the tissue layers, cnidocyte, nematocysts. So this is some key points to remember the hydra's anatomy. First, hydra have nematocysts containing cnidocytes, which usually called sting cells. The epidermis cells of hydra is called epitheliomuscular cells, as it contains muscle fiber. Next, the body of hydra can extend or contract. Next, hydra has tentacles that surround the mouth that can reach out and grasp its prey. The sensory cell of an hydra at the epidermis also make contact with a nerve net that will allow transmission of impulses several directions at once. This is in order for hydra to control all tentacles at once to grasp the prey. So this is some comparison of cnidarians and ctenophore. First, in the terms of catching prey, cnidarians use cnidocytes, while ctenophore use coloblasts. While in habitat, cnidarians usually found in freshwater and marine water, while ctenophore usually found in marine water only. Cnidarians special features are cnidarians have cnidocytes to sting prey. Ctenophore have coloblasts to grasp its prey, and some ctenophore also have bioluminescent to produce light. Okay. While in digestive system, cnidarians have incomplete digestive system, while ctenophore have complete digestive system.